Log on, tune in, find out. Another good idea from Cambridge.
So the next talk, it's a great pleasure to introduce Professor John Bryant, who's from Exeter, and has been there, I think, for 23 years. 22 years.
22 and a half.
Okay, well, 22 is good.
Yeah.
Good enough for a physicist.
The big scale of things.
Much better.
Yes.
And he's written recently, he's professor there, but he's written recently a book with John Searle called Life in Our Hands, and we have that on the bookstall at the back at reduced price, and indeed there are many other books too, so do make use of those in the breaks and so on. But John was for a number of years president of the Christians in Science group in Britain, so he's been thinking and writing and talking about these relationships between science and faith for a good number of years now, and it's a great privilege to have him today to come and talk about genetic engineering and his perspective on that from a scientific and religious perspective. So, John, over to you.
So let me say a little bit about my background in this area. I, until just a couple of years ago, was actively involved in research in molecular biology, and over that time, most of my research has been concerned with the very stuff of life, DNA, and its replication, and the genes that regulate it. So I'm experienced actually handling these things in the lab, and I'm going to say a little bit more about that experience when we come on to thinking about stem cells in my next talk. I've also been responsible for introducing into a mainstream British university the very first bioethics course for undergraduates in biology. Other bioethics courses were available, mostly for philosophy and sociology students, but ours was one of the very first mainstream courses in biology. There may be one or two others who preceded us, but we were out there in the forefront. And that's led me to become the Higher Education Academy's advisor on teaching ethics to bioscience students, along with my two colleagues, Chris Wilmot at Leicester and Andy Bond at Westminster. And I find it interesting that the Higher Education Academy saw fit in its wisdom to appoint 3 Christian guys to be their advisory group on this. Not that they knew at the time, but we did. So this is an area that I've been thinking about, obviously professionally, for a long time, and the ethical issues are becoming more and more to the fore as we become more and more adept in working on genes and handling genes over particularly the last 25 years, maybe 30 years. So the first question I want to ask, and I'm very aware that there are non-medics and non-biologists here, so there's going to be a time for chilling out if you're a medic or a biologist, just sort of thinking, yeah, I know all this stuff. But I want to have a little think about our genes, not the plant genes that I've been working with professionally, and what they do. And then I'm going to think about how we might select our genes, which is a form of engineering, selecting genotypes. I also want to think about how we might directly modify our genetic makeup. And I want to just throw in, towards the end of the talk, some thoughts on eugenics and the eugenic movement, which was very strong here in Cambridge in the 1930s, for example, and ask whether any current practices might be classified as eugenics in any way. So, what do genes do? I'm going to just quickly put up a whole series of characters who've got particular talents and expertises, and just ask whether we think that genes have got anything to do with that. Hamilton's not doing as well this season. A very fine runner. Sorry she's not running tomorrow. Some old runners. Also very fine. Fine poet. A fiddler. An Exeter graduate alumna. And these characters here, this mutant here on the left, what's in these genes? I put that one up because this is a cheerleading group at a university where we have a student exchange program in South Carolina, which I ran for 7 years. And the Human Genome Project, as we'll hear later, is throwing up some very interesting data. But the interest of, whoops, come back. The interest lies in how different are these two young women here, and it turns out that genetically they're likely to be as similar as these two here. In other words, the Human Genome Project does not really give us any basis for racism. And in any pair of humans, and I'll keep doing this, any pair of humans of the same sex, the difference between you genetically is likely to be 1% or less. And I say of the same sex because obviously having that extra X chromosome, young women, is going to make a lot of difference to your genetic makeup. But any two women and any two men will differ by 1% or less in their genetic makeup, which is a very interesting feature. But what do the genes do to contribute to these talents, whether it's cheerleading or mutating into this is supposed to be Chanticleer, incidentally, which I think is rather mutated from Chaucer's original, and hence they've got chants on their front, and that's what they chant when the football team is playing. But how many of those talents could we ascribe to the genetic makeup of those individuals we've been looking at? Is it all in our genes? And in fact, what do genes do? Now, if you're Richard Dawkins, and his famous and groundbreaking book, Selfish Gene, is 30 years old, and it's gone through a number of iterations and mutations. If you're Richard Dawkins, you would probably say, unless pushed into a corner, that actually we are subject to our genes in almost everything we do. He does have an escape clause, which we'll look at in a moment, but he's very strong on being a genetic determinist. On the other hand, an equally atheistic writer, John Dupre, who is the professor of sociology of science, philosophy of science, equally atheistic, but is equally sure that we are not genetically determined. Very much of what we are as persons has nothing to do with our genes, and challenges Dawkins. Richard does come back a little bit by saying, well, actually there are these other things, there are these units that are transmitted in some way from brain to brain that actually help us to override our genes, but nevertheless we are, of course, still regulated by these means. Not that there's any evidence for means anywhere. Remember, this is coming from a person who says, I refuse to accept anything for which there is no evidence, i.e. religion. So there's the religion of the means there. Anyway, some partial answers to what our genes do, and we need to think about this in relation to what we might be doing when we start thinking about modifying or selecting human genes. A gene is a coded recipe. That's effectively what it is. It's a recipe written in a biochemical code. And so if you think of our genomes, that's the complete collection of the genes that we each have, divided into a number of chromosomes, 23 and 23 pairs in most of our body cells. And so each of those chromosomes is like a book, and the genome is the library of coded recipes. And so, for example, as if I was giving this talk after lunch, I would tell you about the gene that contains the recipe for insulin, and that particular gene would be very active after lunch as we start to regulate our blood sugars after the carbohydrates that will be an inevitable part of what we eat. So that the insulin gene is effectively a recipe telling cells how to make insulin. But are we just the product of our genes? None of the research will support that finally. And even Richard Dawkins, an ardent reductionist, has to actually escape that conclusion by introducing this topic of means. And so we do need to be aware of the genes are us fallacy, another version of the naturalistic fallacy, that genes are us, that we've defined our genotype and therefore we know who we are and what we are. So, and again, a bit technical now, we're going to think about genes and DNA. Everybody must have their favourite molecule, and mine is DNA. It's obviously been my bread and butter for a number of years. It is the most wonderful molecule in the world. Without it, none of us would be here. How does that relate to these genes, these recipes I've been talking about? Well, a particular stretch of the string of DNA is a gene. It's got a specific instruction or recipe in it. And if you like, the chromosome is therefore a whole string of these recipes just joined end to end with gaps between them. So if we were to stretch out a piece of DNA across the floor here, and we could walk down it, we would find a gene, a recipe, and then there'd be a gap, and then there'd be more. And in the old days, the early days of molecular biology, before we had pinpointed where genes were, we used to be walking the chromosomes to find them, chromosome walking, dividing into bits and looking at each bit. And there's a tiny, tiny part of a gene, and here are the initial letters of the building blocks. I want you to think of the building blocks as like popper beads, poppered end to end to end to end. So we can make a huge long string of these proper beats, and I've put 4 different colours to represent the 4 different types of building block. And the recipe is written in the order in which these building blocks are organised. That is the code. And those of you who know biology will recognise that here's the start of the recipe coding sequence right there. And this is a tiny, tiny part of a gene which will go on for hundreds or thousands, or in some cases tens of thousands of units, running right the way down into North Cambridge from here. And a wonderful thing for us biologists is that every single living thing on the planet reads the code the same way. And if ever you wanted evidence for a certain single origin for life on Earth, the universal genetic code, or life as we know it, I should say, on Earth, who knows what happened with earlier, maybe experimental forms. Life as we know it is based on the code in DNA. And if the code were not universal, genetic manipulation, genetic engineering would not work. We would not find it possible to take the insulin gene from a mammal, from a human, and put it into a bacterium. But because the code is universal, the bacterium, having never seen that gene before, nevertheless reads it and makes the stuff. So the universality of the code and the oneness of living things. And just to go slightly more technical, here's a gene sort of working. And again, for the non-biologist, let me say that all organisms keep their genes in a particular location, and in all organisms above the level of bacteria, that's actually enclosed in an envelope. And a particular location in our cells is called the nucleus, as it is in a plant or yeast. I keep doing this. And so there's this coding sequence, that's the recipe. There are punctuations, and there are controlling elements that tell the gene when to switch on and off. But the genes are kept in that compartment in the cell, away from harm's way to some extent. And what happens is that working copies are made, called messengers, and it's the working copies from which the recipe is read. And this is, shall we say, insulin, or human growth hormone, or a digestive enzyme, or whatever. It's one of the working molecules of the cell. And the function of this stage is at least twofold. Firstly, it keeps the genes out of harm's way, tucked away, because out there in the cell it's a tough life. It's a lot of rough and tumble going on out there. And the working copies are expendable, they're turnoverable, because you can always go back and read the gene again. But also it enables the cell to have many copies of the recipe out there working. You can copy this many, many times, and so you can have a handful of copies up to several hundred to several thousand out there, depending on how much of this stuff you want to make. And so that's how genes work. And when we are talking about genetic engineering, that is to modify the genetic makeup of an organism, this is what we're asking to happen. We take a gene from one location, a human for example, and put it in another location, a bacterial cell, and we're asking the bacterial cell to behave, to read the gene as one of its own, and to do this with it. Okay, so let's think a little bit about I want to talk about selecting our genes, and the background for this is that much of human genetics over the years has been based around a medical approach, observing that particular conditions, and this has been known for a long time, particular conditions run in families. They were heritable traits. Even way back in the pre-Christian era, it was realized that particular conditions may run in families, and you could plot particular traits that were rather unusual. An example might be there's a mutation amongst the dark-haired people of the Mediterranean basin, which actually gives them much redder hair, and that would have been visible as a trait going through families. Steve Jones, the professor of genetics at University College, tells a lovely story of a rabbi in 8200, in the Christian era, but a long time ago, who after circumcising a little boy on the 8th day, according to the usual rites, the little boy bled to death. And a subsequently born little boy in the same family was circumcised and also bled to death. And the rabbi wisely wondered whether this was a familial trait. He didn't understand how that might have arisen, but nevertheless, he then exonerated any other boy in the family any future sons and male cousins from being circumcised. And what he didn't know was that he discovered X-linked haemophilia, which we now know as Queen Victoria's gene. But if we then go back to the burst of human medical genetics that really started to gather pace after the Second World War, we've now come to the point where there are somewhere around 4,500, it might be slightly more, single gene disorders. That is where a change, a mutation in one gene has caused a heritable medical condition. Some of these are extraordinarily rare and others are much more common. Some are rare in national terms. I was giving a talk on genetics and ethics to a school in Sussex in March a couple of years ago, and a young woman came up to me. She was in year 13 in the school, the last year at school, and she told me she had a genetic condition which affected her muscle development, and she was one of only 3 known sufferers from that condition in Britain. And of course, with only 3 known sufferers, there's no money going to that because the pharmaceutical companies want large numbers of patients. There's no patient support group. There aren't enough people. And so, there's an interesting thing about discrimination against rare diseases, the orphan diseases. But we're also beginning to understand, as a result of these studies and the Human Genome Project, that there are many, many conditions which are at least partly contributed to biogenetics. That the genes give us some sort of susceptibility to a condition. Some of those susceptibilities are very high. Breast cancer 1 mutation gives you an approximately 70 to 80% lifetime chance of having breast cancer. The colon cancer mutation, which is related, gives you about a 60 to 70% chance of having colon cancer sometime in your life. And there are other susceptibilities. That it is true that some people are genetically more prone to viral infections because of the effect of genetics on their immune system. So I'm going to talk about some of these genetic conditions, and I put this little code in DNA out there to remind you that here we are talking about identifiable single base changes or identifiable deletions from chromosomes, things that we can pinpoint and are related directly to the condition we're talking about. And recessive illnesses are where the genetic trait the disease trait only appears in you if both the copies, the one you got from mum and the one you got from dad, both copies are mutated, recessive. So in these cases, one good copy can override the bad copy, and the commonest genetic condition in Britain, it's regarded as a disease. There are 1 or 2 others that are commoner than this, but one is cystic fibrosis. And the carrier frequency, that is the number of people who've got 1 mutated copy in Britain as a whole, is about 1 in 25. And the nearer you get to the southern Celtic fringes, Wales, Cornwall, Brittany, the higher that carrier frequency goes up. So here we've got almost certainly a genetic mutation that arose in a Celtic population. We're still seeing founder effect there. The carrier frequency in parts of Brittany and in Cornwall is about 1 in 22. And since I'm from Breton stock, doubtless that involves my heritage as well. So if you work out the chances on a 1 in 25 of having a baby with another carrier, and then realise that each conception has a 1 in 4 chance of having both copies of the gene, you can see why cystic fibrosis is the commonest inherited condition in Britain. And then if you are of Afro-Caribbean origin, then your most common mutation is sickle cell anemia, and of course that's very important in molecular biology because it was the first protein for where the mutation was identified, and it was very exciting being here as a student in Cambridge in the 60s while all this was happening. And it's also very interesting because if you've got 1 copy of the gene, your red cells are not quite perfect. The recessive condition is not 100% good, but you're protected against malaria, and that gives us what's called heterozygote, other zygote. We've got 2 copies of the gene there, heterozygote advantage. And then we have those dominant conditions where actually you only need 1 mutated copy and you've got it. And Huntington's is one of the most unpleasant of these conditions that we see, which is a neurodegenerative disease that basically you just sort of fall to pieces in your 40s onwards. And I've seen cases of Huntington in South Wales where it seems particularly prevalent for reasons we don't understand. Maybe Tim can tell us what that should be. And then there's those X-linked diseases. These are recessive diseases, but because women have got 2 X chromosomes, unless they're unfortunate enough to inherit 2 faulty X's, they don't have the disease. But the men, guys, we have a problem here because we have this little pathetic Y chromosome. Now, it's very important to us because obviously the testis determining factor is quite an important feature of our life. And the other things like football and air guitar are also equally important. but when it comes to balancing, a recessive mutation on the X chromosome, it doesn't work. And Queen Victoria was carrying the mutation, and if you look at the male progeny from her, you can see many of them had haemophilia. Now, that's the motivation for a lot of studies on human genetics, is the idea that we can alleviate suffering by beginning to understand more about the illness themselves and how they work and what the inheritance patterns are. And the Human Genome Project was a way of coordinating what was a very disparate set of research activities going right across the developed world. If you read 1 or 2 of the American books, you get the impression that America invented human genetics and was the sole perpetrator of the Human Genome Project. It's not so, of course. I mean, back in the early 80s, the late 80s, I appointed to my department a young woman working on sequencing genes on chromosome 19 in relation to immune deficiency. But the Human Genome Project drew all these disparate efforts together. The motivation of determining that base sequence, the sequence of those building blocks, right the way through the human genome, in order to understand what happens when genes are mutated, what happens when genes go wrong in their working life, for example, to cause a cancer, that sort of thing. and 10 years after its initiation, because there was already quite a strong foundation, we weren't talking about starting from ground state, we were talking about labs all over the world already doing it. Progress is quite rapid. And there was a sort of approximate draft, and those of us who write a lot know, the first draft is often actually kind of, thank goodness that's out the way, but it does need tinkering with. And the tinkering then gave us a more or less complete copy of the average human genome by 2003. And of course, subsequently, we've been looking at divergences from that and looking at the genomes of different groups. Ethically, if we want to introduce the first ethical issue here, there was a lot of tension between the publicly funded labs working this, for example, the Sanger Centre, which is the British Centre for the Human Genome Project, and the privately funded labs that kind of muscled in when they saw dollars as the kind of most major motivation. If you understand genes, we might be able to develop treatments, and that way we'll be able to make us lots and lots and lots of money. And so one company, led by Craig Venter, sometimes we call him Darth Venter, because he seemed to come over from the dark side. His company bought 300 DNA sequencing machines about halfway through the project and partially sequenced a large number of genes. And the private corporations involved here very much wanted, on the whole, to place the sequences under patent protection, so protected as intellectual property. They don't own the genes, but they own the right to exploit them, whereas the publicly funded labs, or the charity-funded labs, very much wanted those sequences to be in the public domain, available for health services that were provided by the nation or by private organisations. And in the main, actually, the publicly funded labs won the day, although there are significant numbers of human genes that have been patented, and that has affected the availability of them for use in clinical diagnosis. For the biologists here, you'll be aware that we reckon that to run a medium-sized mammal, you need about 100,000 genes at least, if you look at the number of proteins we've got. And we've only got 25,600, which is kind of curious. How do we manage with so few? And many genes must be more complicated in their mode of action than we think. And we talk about, in the world of molecular biology, we talk about moonlighting proteins, that is gene products, the products of those recipes that do more than one job. And in my lab some 12 years ago, we stumbled on one of these genes with 2 jobs, and it kept me in grant funding until the time I decided to give up experimental work a couple of years ago. So there are moonlighting genes. And for me, the sense of wonder about our biology is very much increased by that. Genes are wonderful anyway. You know, the creator's done a brilliant job here, but they're even more wonderful because of the complexity of what they must do. And so the writing of the final draft was really the end of the beginning, and we can now really start to get to work. And we can get to work also in our medical applications of these things. And so what I want to think about is how might we use, in a clinical context, how might we use that knowledge we've now got, identifying the changes that occur to cause an inherited genetic condition, how might we use that knowledge? And the first thing that we can do is that we can start to identify those changes in individuals. We suspect that an individual might possess a mutated gene, whether a single copy or 2 copies. We can start to test that out. If for example, you've got, if the women have got breast cancer, it seems to be a familial trait, you don't need to wait till it develops. It's a symptomless test. DNA is DNA, and you don't need to wait until symptoms develop. You can test the person long before there's any signs of illness. And so a lot of genetic testing is pre-symptomatic, but a lot of it will confirm another medical diagnosis. So adults can be tested, but the adults maybe want to be tested for reasons that they think they might get something, or new symptoms have risen late in life that are only explicable in terms of a known genetic illness. You can test children again. For example, if a lad starts to develop muscular weakness, and we might suspect Duchenne muscular dystrophy, you can test the child. You can test newborn babies, and indeed, for many, many years in Britain, newborn babies have been tested for genetic illness, phenylketonuria, but by chemical method. And now you can test that by genetic method. And in different local authorities, different health authorities across the country, different diseases are routinely tested for in newborns. So if you come from the Greek Cypriot community in London, for example, automatically thalassemia will be included. Cystic fibrosis is automatically included now for all newborns. So we're beginning to see a situation where we're applying genetic tests as pre-diagnostic or as confirmatory diagnostic tests, even with babies. And of course the ultimate is that lovely film, well, interesting, disturbing film Gattaca, which I think everybody should look at. Gattaca is simply made up of the 4 letters of the DNA bases. But it concerns the efforts of somebody who had been born by the normal conventional method, not by IVF, where all the other children had been conceived. He was conceived as a result of the normal fun methodology, and his parents were students, and it was in the back of the car. And so he escaped being selected as an embryo, but he was tested as a baby, and of course his barcode showed that he was prone to sudden adult death through heart disease. His efforts were to con the authorities to let him go on the space shuttle. And quite interestingly, a civil servant at the Department of Health asked 2 of the government's advisory committees to look at whether we should barcode all our babies for health-related genetic issues. And they rejected it on the grounds of 1, practicality and expense, and 2, well, the possibility for misuse of such information is immense. And we need to society needs to think about that before we go down that road. But it has been suggested at government level. We can test fetuses, and fetal diagnosis of genetic illness has been around for some 40 years. You can get fetal cells as early as 12 or 13 weeks if you know what you're doing from the chorion villus. And of course that raises issues about, well, if I understand that my child has got this genetic condition, what do I do about it? Do I terminate pregnancy? Difficult decisions for prospective parents. And more recently, since the late 80s, we've been able to test pre-implantation embryos, that is embryos that have been generated by IVF outside the human body. And before they're placed back into the woman's womb to try to establish pregnancy, they're tested at a very early stage. I'll pass that by. And the reason that works is because of the lack of determinacy developmental-wise of these early human embryos. These are wonderful pictures of early human embryos, and I have to thank the journal, The Biochemist, for allowing me to use them for educational purposes. The newly fertilized human egg, going through the cleavage stages, the cell division stages, and this structure will come to much in the next talk. But this normally happens, of course, in the fallopian tube, this series of divisions as the fertilized egg is moving down towards the uterus. But at this particular stage, those cells have no idea what they're going to become. There is no polarity, front, back, up, down, left or right at this stage. And as I've said before, that's very different. If you were a fruit fly, and I can't see any of you, it would have been very different. You know, the fruit fly egg has got a front and a back and a left and a right and an up and a down, whereas that doesn't happen in humans until pregnancy is established. But it means that you can take one of these cells out, and because of the powerful methodologies of amplifying, copying DNA, we can then test that cell for the mutation, genetic change we're looking for. And that's pre-implantation genetic diagnosis. And people who are at risk might decide, well, I wouldn't want to abort a fetus because that's a human growing in my womb. But these embryos outside the body, they might consider, would be less ethically significant. And so I might want to discard the ones that are working. And that actually happens, and we'll think a little bit more about that in a few moments. That if you know there's a serious genetic condition in your family, you can now elect to go for IVF and have a disease-free embryo. But also, more controversially, of course, we've had the situation recently where an older child, for example, needing a stem cell transplant that needs to be immunologically compatible, the parents can select an embryo out by IVF that could be a donor for that child who's already been born. The so-called saviour sibling scenario, that you have a child to be a saviour for another child already born. The number of cases is very small. It's a handful of cases, both in the UK and in the USA, but they always attract a lot of attention. And if you want to read a beautiful and moving novel about saviour siblings, just go and get Jodie Picoult's My Sister's Keeper. It's set about 10 years in the future, but medically and scientifically, she's done her research well, and it talks about the downside of being a saviour sibling. And all this is regulated by the Human Fertilization and Embryology Authority, and they have included in the list of illnesses that we're allowed to test for those late onset conditions such as colon cancer and breast cancer and Huntington's, and they're also increasingly looking at conditions which really just give you a high susceptibility, like those cancers I just mentioned. Hypercholesterolemia, genetically elevated high levels of cholesterol in the bloodstream, which if you've got 2 copies of the gene is a strong indication of death in childhood, is the most recent added to the list. So I want to tell you about Chloe, that had we got access to the internet in here, I could have actually shown you the video, but Chloe is a baby who was born by IVF. But the reason she was born by IVF is because her dad, who lives in Chicago, has in his family colon cancer gene, and the colon cancer gene had devastated different bits of his family. It's a dominant mutation, so you only need 1 copy. And Chad and his wife Colby decided that they wanted to have babies that were not going to be under threat from that mutation, so they elected to have IVF and then genetic selection of the embryos to reject those that were carrying the mutation. And the video tells beautifully the story of their discussions and the fact they put it to their family members and how one of their church groups objected strongly, and we could get to talk to the doctor who raises issues about the slippery slope and how far down we should go. The interesting thing about Chloe is the condition being tested for is cancer of the colon. The frequency with which somebody with the gene will actually have the illness, which develops in sort of 40s, 45, 50, and this will be a mutation that Tim is very familiar with. The particular mutation being tested for gives the carrier of that mutation about a 60% likelihood of having the condition. And so 40% of the people with the condition, or with the gene, don't have the condition. And then in American medicine, if the condition is detected early enough, there's now something like a 70% 5-year remission rate, cure, shall we say. So we've got a late onset gene, which does not give you a 100% chance of getting the illness, and for which now, with good medical support, there is increasingly successful. And the doctor starts talking about, how far down that line should we go? How serious a condition does it have to be? And how high should the probability be before we start saying, well, are we going too far with our intervention? It's an interesting case. But let's look at what's around out there. This is the availability of direct genetic tests. And across the world, there's 1,258 genes as of a couple of weeks ago in clinical use. Now, I need to say that the term clinical use here is a bit sloppy, because once the gene sequence is out there in the public domain, anybody can use it. And a lot of the genetic testing services, and we're not just talking about the USA where medicine is commercially funded, we're talking about Britain as well. A lot of the genetic testing services are not actually taking place in a clinical context, even though they're being used to detect a clinical condition. And often the people offering the tests are not skilled in interpreting the results, the probabilities, and so on, to the people who receive them. But if you just want to go to genetic tests and Google those, you'll come across a huge number of companies that are offering tests. And they're offering, for example, genetic assurance, which I assume is a secular version of blessed assurance, or something. And the number's growing. Now, in Britain, the situation is that quite a large number of these tests are available in the clinical context, as well as being commercially available. And the HFEA is the organisation that decides whether or not embryos should be testable. And the last time I spoke to some of the HFEA, there's some 70-odd conditions in Britain for which the NHS will pay for you to have an IVF embryo tested. And so we're beginning to be able to select the genes of future generations based here on medical grounds. It's becoming more and more technical. The question then, and this is I'm going to throw questions at you rather than tell you what I think. The question then becomes is how far should we be allowed to go along this line? Should we start selecting non-medical things with our embryos? Just a thought. And so here's a few more issues. Is medicine becoming too geneticized? The genes have spoken and nothing else matters. Are we beginning to narrow our concepts of what is normal? We start, for example, trying to eliminate achondroplasia, the short stature condition. Tom Shakespeare is very, very lucid about this, and he has achondroplasia, the professor of medical ethics at Newcastle University. Will we begin to discriminate or marginalise? Will we slip back to reductionism? If the genes are this gene is going to do this to me, well, maybe all genes have an effect that I can measure. And then we've got these problems, and there are now documented cases in the USA where people are being rejected from insurance. Rejection from employment, well, there may be good reasons for rejecting somebody with a particular genetic condition from employment. And if you're training a pilot to fly planes across the Atlantic, you don't want her dropping dead at the wheel, for example. So there may be really good reasons, and society's going to have to think about this very carefully. Okay, then very quickly for a few minutes, direct modification of our genetic makeup. And we generally modify cells, and this is something I've done many times in yeast cells and plant cells. We generally modify them by taking a small piece of DNA and splicing into it the gene of interest. And then for mammalian cells, this is usually injected into the newly fertilized egg, or for large mammals, such as humans, but we don't do it with humans. So that's by the early embryo, but we can also, for small mammals with a short generation time, like mice, we can use embryonic stem cells. But we can genetically modify mammals, that's the point we're making, with specific genes. And when we talk about genetic modification with humans, we're generally thinking about gene therapy. That's replacing faulty genes or correcting the activity of faulty genes. And it goes into this gene therapy is directed straight at the cells that are most affected by the mutation. So although cystic fibrosis, for example, affects a number of the transporting membranes in the body, the lungs are the most affected. And so for those conditions where we can identify a set of target cells, then we can actually try to correct the faulty gene. And these are the three conditions where there has been the most research. Cystic fibrosis has not been successful. The severe combined immunodeficiency, the boy in the bubble syndrome, the success rate in both London, Great Ormond Street and Paris has been between 90 and 100%, although in Paris and more recently in London there have been side effects reported for a small number of patients. They contracted leukemia-like disease because the incoming gene has inserted near what's called an oncogene, a gene that will switch cancer on. But the success rate of curing the disease has been very, very high. And then very recently, congenital blindness, a gene's been spliced in the cells in the retina, and we're still waiting to see how successful it will be. And then in the States, we've got therapies now that are based on turning genes off, genes that have been activated and cause cancer. RNAi stands for RNA-based inhibition. RNA is that messenger molecule I talked about. But of course, we could do it in a heritable way. My own view about heritable gene therapy is that it probably won't be developed because people will select the embryos instead. If you want to correct a faulty gene in your germline, you'll select the embryo rather than genetically modify the embryo. And in any case, it's currently forbidden. Any form of genetic modification in an embryo you put in the womb is forbidden. But quite interestingly, the public support the idea, but as I say, I think that's mainly because they're not aware of embryo selection. But if you went out in the streets of Cambridge or Exeter or Nottingham or anywhere, on the whole, people think it's a great idea, eliminating a faulty gene from future generations. As I said, I think the fact we can now do pre-implantation diagnosis has removed the pressure of developing germline therapy. But what it hasn't removed the pressure for is the possibility that someday some folk might like to try to, we can select non-medical conditions. We can select the basis for blue versus brown eyes. Let's forget the modified genes. We can do some selection when we're doing PGD. We can do some selection for things like stature, some selection, very multigenic trait. We can do that sort of thing. We can do some selection for colouring. But maybe rather than waiting to see what your batch of embryos turns up with, why don't we just make sure that we have a brown-eyed girl, if you're Van Morrison, and do the genetic modification. And certainly there are people out there who think that genetic modification of humans will be achieved in the first half of the 21st century. And of course, once again, we start wondering whether we can tell the difference between a genuinely medical intervention, which, as I say, I think on the whole is redundant, and a non-medical intervention. And in the case of cosmetic surgery, I've got a student this year who's doing research on attitudes to cosmetic surgery. And it's been very, very clear amongst the people that she's interviewed and the questionnaires she's administered that it's very hard for some people to distinguish between what is really enhancement and what is really therapy. So some women are claiming that breast enlargement is actually therapy because it changes their emotional state, which is interesting. Is that enhancement or therapy? So we have exactly that. Now this slide's coming on backwards. So if we go down that road, and as I say, there are people who believe we will. I was talking with Gregory Stock from UCLA a couple of years ago, who writes a lot about this and talks about the future genetics of our children. He believes that we will go down that road and people will be prepared to pay for it. And then our baby becomes a sort of commodity, and maybe some people with views that, you know, the baby is a gift from God, might feel, well, you know, we're actually kind of trying to bypass what God gives us here. And baby becomes a commodity, becomes my designer handbag, I've got my designer baby. My designer handbag maybe have a tiny dog in it, well, just the baby on the arm instead, kind of skeleton. Now, I want to say that because at the moment we conceive only the possibility, and I didn't mean to use that pun, we think only the possibility of using IVF as the route for this. At the moment we cannot envisage it's going to become mass technology. You know, we're not going to grow great armies of people looking the same. I'm not going to be able to raise a football team that will get Palace back into the Premiership. And I'll be tuning into my radio for results later. No, I can't be listening to the talk. So we're not going to do that. You know, we're not going to design another Paula Radcliffe because of all the elements of non-genetic influence on somebody's life. We can't design a person. And also, as I think Philippa will say tomorrow, that we're much closer to widespread cognitive enhancement than we are to genetic enhancement. Again, one of my other project students this year has been looking at cognitive enhancement, and we are very close to that being, if you like, not necessarily available in a widespread way, but available to those who can afford it. And I think that will become, that will come to us before we get to genetic enhancement.
Can you just explain what cognitive?
Yeah, it means increasing the capacity of your brain in various ways, increasing your attention span, increasing the speed of functioning of the brain, and so on. And some of this is actually funded by the US Department of Defense in the Future Soldier Program. That enables soldiers to think faster and for longer and not to fall asleep, for example.
Is that basically an educational program?
The Future Soldier Program, no, it's not.
Cognitive enhancement.
Well,
is it an educational thing or is it a genetic based thing?
Well, no, it's based on mostly at the moment pharmaceutical interventions, but there's an increasing number of people who talk about transhumanism who are talking about interventions based on welding human to IT devices, for example. The transhumanists. I think Philip is going to talk about them tomorrow. Well, are there religious views about this? You know, I think actually it's quite difficult to categorise. Are there views on the intrinsic rightness and wrongness? Are there, in Neil's terms, are there deontological views on GM? And I think actually, when you look around at the views, they're nearly all consequential, you know, or maybe they're virtue, but it depends on what you're doing with them. You know, what organism are you trying to change? Why are you trying to do it? And we have some examples of the discussion going on. If you're selecting embryos, are you preventing serious illness? Are you destroying life? Is genetic enhancement unacceptable, or is it doing the best for your baby? The atheist philosopher John Harris would argue the latter. That you know, if you're happy to pay for music lessons and for football coaching, why not pay for a gene job instead? And he says that, you know, and I use his words, but for genetic selection now, he says, if a couple want nature or their God, as he puts it, to provide them with a bouncy, curly-headed, brown-eyed girl, another Van Morrison fan there, why don't we make sure they get that, if that's what they want? So it becomes a matter of autonomy and personal choice. And very quickly, some thoughts on eugenics, and I just throw these out because I want you to think about whether any of these practices are moving us in a eugenic direction. Of course, the word eu is the same eu as in euthanasia. Euthanatos is an easy death, or good death rather. Good eugenics is good origins. It doesn't mean, say, you hold your knife and fork properly. It means good genetic origins. And Darwin, who didn't really know anything about genes, but they weren't really kind of discovered until some years after the publication of The Origin of Species, nevertheless wondered whether we could apply evolutionary theory to society. And that goes back to ideas expressed as long ago as the times of Aristotle and Plato, who wanted to have an improved society. And I think it was Plato who said that the best men should have sexual intercourse with the best women as often as possible. And of course he was probably defining his class as the best men, as you might imagine. And then Francis Galton, the first professor of genetics, Darwin's cousin, then formalised eugenics, and particularly encouraging the fitter to breed more extensively. And of course the fitter were the liberal middle classes, the Wedgwoods, the Darwins, the Lovetts, etc. These were the people who were deciding who was the fit and who were not. And the eugenics movements were popular in the UK, but they were quite amateurish, although when I was a student here in the 60s, you could still find in the genetics department some old posters with showing the farmer distributing the grain by the old-fashioned broadcasting method. And the sign underneath said, only broadcast the best seed. They weren't, of course, talking about wheat there. And so those dated back to the 1930s. I suppose if I was really sick, I might say that eugenics is like so many other things that were invented in Britain. Other people did it better, and I would include cricket, football, rugby. But eugenics or so. And if I could just talk briefly about the USA and about Scandinavia, talk with colleagues in the southern states who tell me that, I reckon the number of eugenic deaths in the States is around 20,000, but apparently the number is much nearer 50,000, going right the way through from the 30s to the 50s.
That's right, the latter figure is more accurate.
Yeah, yeah. And often classist and racist elements here, that a judge would decide that this particular young woman was morally feeble, and it turned out she was a black servant working in a white estate, maybe down in Georgia or South Carolina, and she'd been raped by the young master of the house. But she was morally feeble, so she'd been sterilised. And if the baby was a girl that was born as a result of that rape, the baby was sterilised too. The BBC tracked down the daughter in just such a case some years ago and interviewed her. And as I say, some 50,000 eugenic sterilisations were carried out in the States. The most recent eugenic sterilisation, again carried out on the grounds of being morally feeble, was in Scandinavia, in Sweden, in 1974. So, that's pretty darn recent. And so this, and the people making the decisions were, the liberal, educated middle classes in every case. And it just occurs to me from time to time that, if we start working on genetic selection and so on, are we beginning to narrow our idea of what we want in society? Does that have any bearing on the eugenics that was aimed at improving society in a sociological way? And just to wake up those who can see, got a bell there to ring. This comes from an article by Madeline Bunting, a very, very thoughtful columnist in The Guardian, who writes quite frequently about ethical issues arising in different areas of societal practice. And here she's talking about, she looks forward some 35 years to when her daughter and her daughter's husband, because her daughter's quite little at the moment, will be looking at the progress of their child through school and thinking about the pressure there has been for genetic selection of embryos by then, for these multigenic traits that might make the child stronger or fitter, and the fact that they had rejected cognitive enhancement, and that was putting their child under a lot of risk for not doing so well at school. And might there need to be special classes for non-enhanced? And it's out there, genetic enhancement, genetic selection, cognitive enhancement. Some of this will be cleverer and better. And what does that mean for our common humanity? What does this expensive medicine and this expensive enhancement technology mean for the billions of our population who don't have enough to eat and drink, who see their children dying of diseases which in any developed country would be completely preventable? So you've got this kind of balance across the world, and yet there are people like Gregory Stock who are going all out for enhancement and transhumanism. And I think she believes, rightly or wrongly, that we've got to a runaway situation, in which the situation I think that was described by Tim, that if we can do it, then we should do it, because an individual life might be enhanced. It's our right, we live in a very rights-based society here, which I'll say a little bit more about this afternoon, so why should we deny people our rights? But of course it will be the rights of the few. So there is no stop button, and I think that's the situation that we're facing. And as Christian or religious believers, we do have a contribution to make to that debate. But it needs to be a thought-out contribution, not a gung-ho, that's right and that's wrong. There are nuances here that we need to think through. So at that point I'll stop. Oh.
Thank you, John. That leaves us a good half hour again for questions or comments. Who'd like to start?
I always work to instructions.
Yeah, absolutely. Yeah, I think one of the good things of the way we set it up is that there's plenty of opportunity. Anyway.
In terms of genetic selection, especially in embryos, you mentioned earlier on in your talk that for a lot of, you were surprised at the small amount of genes that they found in humans.
Yes.
And that therefore there must be a lot of moonlighting proteins in genes that have several functions.
Absolutely.
So if you're selecting against other genes on the basis of probability, without knowing the full probability of something else happening, how can you say, well, there's 100% chance that we'll remove this disease in this baby, but we don't know the chances of something else happening. So how can you judge anything on that without knowing the full consequences?
In terms of genetic selection, we are specifically talking about those instances where we do make a direct link between the mutation and the condition that ensues. And we must not assume that all our genes are moonlighting. I mean, you'd think that we need to have every gene having 4 functions, but it is a slight paradox that we do know many that have simply a single defined function. I think I'm much more concerned about genetic modification in your case, because what we don't know is whether putting a gene into a complex environment like the mammalian genome, where a significant number of the genes must have multiple functions, will actually in some way change the way the genes work. So it's a much more a problem for genetic modification than it is for genetic selection. I mean, what you're not.
I guess I'm thinking on a long-term scale of changing gene frequencies in population.
Well, you're changing the frequency of that mutation, and if there's what's called heterozygote advantage, in other words, if the single copy of the recessive gene gives you some advantage, then you would lose that. But what you're not doing is you're not losing the wild-type copy from the population, because you're enhancing its frequency. And if there is no heterozygote advantage, it seems to me that is not evolutionarily a problem. There is heterozygote advantage, then you need to find some way of dealing with that.
I mean, just going back to the issue of selection, I remember having a vigorous argument with somebody about this in the late 80s when it was just coming out, and the concept that you're going to be, in an IVF situation, you're going to be doing pre-implantation diagnosis, and you're going to be selecting the healthy embryo is very appealing. But it does raise all sorts of questions, doesn't it, and about what's happening to the other embryos, and the efficiency of IVF as a system anyway.
Yes.
And as you elucidated, what is the limit of this? So, do you feel that as it is now practiced, for severe monogenic disorders, we were arguing about Lesch-Nyhan syndrome, which is a horrible syndrome where children end up sort of going backwards about the age of 6, 7, 8, 9, 10, and all die in relatively early childhood in horrible mental degradation. That these sort of horrible conditions that do occur, do you feel any ethical concern about the rightness of pre-implantation diagnosis for these sort of lethal early childhood conditions from a Christian point of view?
I don't have any ethical concerns at all. But I will say that there are other ways of dealing with the situation you just described. For example, one can deal with some of these conditions which are located in particular groups and communities. One can deal with them in a societal way rather than in a technical way. although some technical intervention is required. The Ashkenazi Jewish folk are particularly prone to Tay-Sachs disease, which is a disease where the sheaths of the nerve cells begin to break down, and so from the age of 7 or so, a child will deteriorate very badly and will quickly die. Now, Tay-Sachs, as I say, is quite prevalent amongst the Ashkenazi Jews, who are strongly represented in the Jewish communities in New York and in Chicago, and of course now back in Israel. And a rabbi in Chicago instituted a program which has been called Eugenics with a Smiling Face. I don't know what the Hebrew for that is, but that's translated out of the Hebrew. Where all young people, prior to the age when they like to have children, are now tested for the Tay-Sachs mutation. And the rabbi keeps the record. It's obviously blinded, so he has to have 2 sets of records. The young people don't know, their families don't know whether they've got the mutation. It's recessive. But if they are planning to marry, and this obviously only works for societies where there's a strong community with a kind of its own ethics and ethos. If the young people are planning to marry, they will go to the rabbi and he will translate his blinded copy into the actual data and say, well, yeah, you two guys, you've both got Tay-Sachs mutation. I say guys in the American sense. Yeah, we haven't yet got to the point where 2 gay men can have a conceived baby by normal means. You two are at risk because if you have children, each one has a 1 in 4 chance of having disease. And so he provides strong advice that they shouldn't marry, or if they marry, they should not have children, which is a tough decision, but the young couple is supported by the community in which they live to make those decisions, and the frequency of the mutation has dropped very, very markedly because in the USA, particularly in Chicago and New York. So that's a societal way of living. And we see a similar situation with thalassemia in Cyprus, where the Orthodox Church is encouraging a similar program, although the medical authorities in Cyprus will have freely available abortion for thalassemia. So there are other ways of dealing with it. But in terms of your actual question, given that we don't have that kind of strong community, I'm actually okay about selection against these very, very nasty illnesses. But how far down that track of 70 that the HFA allow will have gone, I do not know. I mean, some of them are really very unpleasant, but some of them seem to me to be much milder and can be dealt with by much more normal medicine.
Can I just press you a bit on the concept that something is bad? Because you've always said these are diseases we're discriminating against in PGD. And I know of cases where people with some, we would consider it an abnormality, say 2 blind people who are married, actually want to have a blind child, because that is normality too.
Yes, indeed.
How do we deal with
well, the people who are deaf have actually been even more vocal about that. They do not want to select against genetic deafness, and I think that's...
Well, not only not select against it, but select for it.
Yes, I find that one quite tricky, because with the best will in the world, a deaf or a blind person is at a disadvantage however much we take care that we don't discriminate. It's at a disadvantage, and the question that you need to ask the parents is that, are you happy for your child to have the same disadvantage in this area as you do? I wouldn't want to comment.
Well, who's going to decide what's advantageous and what's disadvantageous?
Well, that's the other thing, that it's very hard for us to decide the quality of somebody else's life, particularly if that life is yet to come. But there are conditions where the life of the child is so difficult, as Tim has alluded to, that I would have no hesitation in saying genetic selection there is okay. Deafness and blindness is a different matter.
Yeah, that's why I raised it. You know, this is a tricky issue, isn't it? Are we the liberal middle classes again that are imposing our will on others?
The selection for deafness, raises, your antennae start to go up, and what we're asking is the society should accommodate that request. And I honestly don't know whether society should accommodate such requests. I do not know.
I was going to say, I thought that just now society won't allow a deaf couple to not screen at all. That not
Yeah, they will
correct?
allow it on screen, of course. Yeah.
No, I thought the recent case with, he's got a funny name, Tomato Lici, and his, that's my name, Tomato Lici and his partner, I thought they'd gone through IVF and then had said, well, we don't want the child
Yeah,
to
well,
screen.
I think they were misrepresenting the current position.
Right, that's what I wondered.
They're not forced to have genetic screening.
Because I thought that's ridiculous. I thought, well, that is ridiculous. It's very, it's one thing to say screening for deafness, sure, there's a kind of argument there. But to then impose our screening...
No, I heard the interview, and they misrepresented what the
Right,
current situation
okay.
is. They're not forced to select for anything.
Yes, let's go to.
Well, I sort of have all the hesitation that you have about selecting for deafness. What's fascinating to me about that, though, is that you have a community which actually wants it. You know, if you put your Jewish community on the one hand,
Yes.
the way that they will support some of the people there, one assumes that the deaf community will support these children. Now, I mean, we who are outside the deaf community look on this and think, oh, this is awful, because you are actually bringing about a disability. But for them, it's not. And I find this an extraordinarily difficult one, where, in a sense, all our normal ways of looking at things seem to actually collide.
Indeed, and we had a profoundly deaf student come through our pre-program about 10 years ago. It was very, very tough for him. It was quite tough for us, actually, but it was very, very tough for him. But in that good degree, it would have been interesting had one had the social situation in which it was appropriate to talk to him about whether he'd want his children to be deaf or not. Michael's been waving his arm.
Yes,
But
Michael's
it's
turn.
just like lots of people, because it's something many of us have thought about, and I'm not sure may have mine, but a bit more. The argument that it's wrong to permit parents who are profoundly deaf to choose to have children who are profoundly deaf who could otherwise have been hearing, because those children are disadvantaged, is a very difficult argument
It is
to
very
sustain.
hard. Yeah.
And there are many reasons why it's difficult to sustain, one of which is it appears to allow a great many other things to be permitted
Yes.
that we don't want to permit on the grounds that if we did X, something, a person results who's disadvantaged. I mean, there are many cultures where being a woman is disadvantaged, it's a disadvantaged state. And then, of course, disability studies now almost seems to reach complete consensus that the concept of an individual being disabled doesn't mean anything. It's the relationship an individual has with the society, with others. So I always like Dan Keating's quote about basketball, why don't they just lower the hoops?
You might make easy play basketball.
And you mentioned Tom Shakespeare.
Tom Shakespeare, yes, exactly.
Tom himself, of course, is just amazing and wonderful at saying that he doesn't suffer from achondroplasia, but sometimes he suffers from equals.
That's right, which is why I will not list achondroplasia, as it is in some textbooks, as a genetic illness. It's a genetically caused condition, but certainly he's quite happy. But I used to agree with Mary Warnock that the slippery slope is not a valid ethical concept. But actually, the more I think about genetic selection and that sort of thing, I think the more I have to accept that in some areas there is a slippery slope, because there's a continuum between those illnesses such as Tim described and conditions at the other end, which some parents might want to avoid because they want a perfect child. And somewhere down there, each of us reaches a point where we're going to dig the ice axe in. So I'm not going to go down here any further.
And actually, we've already passed that condition in many ways.
Well, I think, yes.
You know, the situation you talked about colon cancer, which I see regularly, that particular genetic
Exactly, yes.
disposition, where you can screen very simply and avoid the problem.
Yeah.
And so this is not even a lethal condition, really.
No, and that's the point that's made by the doctor in the discussion. And I said it's a beautiful video of the whole process, but the doctor keeps making the point that here is a condition which is effectively early detectable and dealt with.
Yeah.
Just before we go to the next question, just give some of us a clue as to what to Google to get that. Do we do Chloe's story?
No. What you do is you go to the New York Times website,
Right.
and you put in into the archive search, you put in embryo screening video.
Okay.
And it lasts 6 minutes, and I use it a lot in class. In fact, I showed it at the Faraday back ago, was it in November? I can't remember. I come so often.
It'll be on our website.
Yes. It probably won't be on your website because they won't let you do it.
You're right.
Otherwise, I'd have had it in my PowerPoint.
It might.
You're allowed to scream something off the room.
Yeah, yeah.
Save your siblings.
Save your siblings, yes.
The one issue I have with saving your siblings and the ethics of saving your siblings is not so much the desire for the creation of an embryo that will save a child, and it's all very laudable, but the issue that I have is the ethics of what do you do with the embryos that are normal, that are not capable of saving the child,
Yeah.
it's a huge dilemma for me.
That Save Your Siblings focuses very specific on that issue. You know, it's all very well doing genetic selection, where according to what you believe is a serious condition, you are preventing the birth of babies with a serious condition. When it comes to Save Your Siblings, at least some of those embryos will be healthy and could have established a pregnancy. And if you like, spare embryos are being thrown away. Now, in IVF, anyway, many spare embryos are discarded, and many of those are healthy. So in a sense, we're not raising a new condition, we're just focusing on what we think is worth keeping.
But the issue that I have is that surely, yeah, we do, IVF does discard embryos.
It
But
does.
in this case, we are creating embryos that we intend to discard that don't meet certain criteria. So we're definitely selecting.
Indeed, and some people are unhappy with that, and others are completely happy with it. And as I say, it's not my job to tell you where I'm standing here, but these are things for you to think about.
Can you just tell us a statistic which always amazes me for rejection of fertilizer?
Well, yeah, different
In
different clinicians come up with different figures, but I mean, the most recent one I heard from Lord Winston was that in a natural fertilisation, the chances of establishing a pregnancy from that are 20%.
1 in 5.
1 in 5. So it does mean, but of course the body's got, if you like, a quality control system. The reason that some of those early embryos will not establish a pregnancy is because the body's quality control system is working on the zygote, maybe even worked on the oocyte, the egg first. So the body's imposing some sort of control, but others, they don't establish pregnancy for reasons we just do not know.
Yeah. But the statistics for human IVF are not dissimilar.
No, I mean, people talk about, we're getting a success rate of up to 30% in IVF clinics, but we're not talking about individual embryos here. We're talking about a round of IVF, which will have generated in the current system. The new MARD system is slightly different. Will have generated in the current system between 8 and 12 embryos per couple, and so that's a 30% success rate based on a large number of embryos, of which 2 will have been implanted.
Well, when we begin to play God, as it were, not to use the term in the trivial sense as we did earlier.
Yeah, I don't like the term actually,
right. But forget all the genetic modification technology. It seems to me that there are already profound ethical questions raised by what we could do with older technology, like ultrasound and amniocentesis.
Mm-hmm.
For example, I was in China for 4 weeks this fall. There are major cities in China with over 1 million people in them, where the ratio of young men between 20 and 30 to young women between 20 and 30 is 130 to 100 or more. And of course, the reasons for that are obvious. And so, do you see any difference from a moral perspective between selection of embryos based upon gender than a selection of embryos based upon one of these minor genetic conditions?
No, it is very interesting that one of the things the HFEA forbids is the selection of embryos based on sex. I was in China for 2 weeks last year, and the little empress syndrome that you portray is very, very real. There are cities in Britain where the obstetrician will not tell you the sex of your baby after the scan. Because of predomination in particular communities to want to abort female fetuses. So my elder daughter-in-law was not given the option of knowing the sex of her 2 little boys before they were born, because a significant proportion of people who went to the hospital would have wanted to abort female fetuses.
Mhm.
So it happens in Britain as well as China. I myself would reject strongly the idea that we should be allowed to select the sex of our baby, unless there was a very good reason that a male baby will have this condition. Duchenne muscular dystrophy would be an example. So that would be, if you like, based on a genetic illness condition, not on the sex. But I would reject that. But that's my personal standing on that one. So the HFA is so strong on this that when a family called the Masterson very tragically lost their only daughter in a bonfire accident some years ago in Scotland, they had 4 boys, and the youngest was a little girl, clearly the pride and joy of the late baby. And they said they wanted to try for another baby to replace the little girl with another little girl. They were getting on a bit. The wife was in her 40s, and so IVF was less likely to work for them. The HFA anyway refused them, so they went to Italy, where until very recently the laws were very liberal. They've been tightened up considerably in the last 2 years, and they had IVF, and after several rounds, the only viable embryo that was generated was male. So they were unsuccessful in replacing the little girl, and they donated the viable embryo to an infertile couple. So it's out. You can go and find it. And it was a very tragic case, but as the HFA said, hard cases make bad law, and so they wouldn't shift even on that one. And I think I agree with them that gender, or as I prefer as biologists say, sex selection, I think should be prohibited except when there's a very specific other reason for the selection of the sex.
But you see, that's our Western ethic, if you want to take a secular point of view on this. That's our Western ethic coming in vis-a-vis the ethic of another culture, which might view being female as a disability, as has already been suggested here today. I'm not sure I see a difference between that case and a lot of these other genetic cases. In a few cases, these extreme cases, I could be persuaded. But in a large majority of these cases, it would be difficult to persuade me.
Yeah, so you're at one end of that slope that I was talking about. You've dug your ice axe in just below case number 5. Other people are down there below case number 70. Fine. I mean, that's the position you take. It's very interesting that in India, which is a society in which traditionally women have had more menial roles, India is changing very fast. And now in India, the doctors are not allowed to tell you the sex of the baby after a scan. If there's a lot of pressure from the parents, they may make sort of obtuse remarks like, go and paint the nursery pink, which might in itself be a sexist remark, but you know what I mean. But they're not allowed actually to formally tell because, again, of the tradition of wanting males. But India is changing fast, and they decided that for their society that they don't want to encourage the deselection of female fetuses. So things do change.
Can I press you on the Christian perspective, make a Christian comment specifically, that if we are made in God's image, and in the fullness of time, if you're a Christian, there'll be a new creation, you'll have a new body which is free of any of these imperfections, if you like. Should we view some disability now as strongly as we do? Because for eternity, we might be killing a person that would have an eternal life.
As we've already indicated, what is for some people a disability is for other people just being differently abled. And I think that's, and Michael made the point very, very clearly there. And I think we have no right to say who's disabled and who's not disabled. I'm specifically talking here about, when I express support for genetic selection, I'm talking about illnesses which are very serious and which lead to a short, nasty British life. I am unhappy about the selection against these late onset conditions for which there is good medical intervention. But I think when it comes to a specific Christian position, we cannot impose that in a society which does not, on the whole, accept our views. You know, we can't impose what I would call ethical imperialism. We can go and argue our case in the marketplace. We can exhibit all the Christian virtues that Neil talked so brilliantly about last night. And different people will apply those virtues in different ways. So that's another thing, that even Christians who are thoughtful and have read the stuff and are trying to act ethically will still reach different conclusions. That will become very apparent when we talk about stem cells this afternoon. So we can't impose a Christian view because there isn't a single one. But on the other hand, I think we can argue for treating other human beings who are here with compassion and integrity as our equals, as children of God, as made in the image of God, and as deserving to have a life that is as fulfilled as we can provide for them. That's for people who are already.
Yes, I mean, what prompted this, and then we'll go to Dennis, was at the end of John Wyatt's lecture that I mentioned just earlier. He gave the case of a little girl who actually lived only 4 weeks, I think, and it was obvious she was going to, but they still did everything to save her life and so on. And he said this little girl's life had a profound influence on an enormous number of people. And you know, if she's a Christian, she's living for eternity anyway. So, you know, should we be selecting against that possibility?
Well, we're in a very tough area here. Some members of our house group there, and therefore we had a lot of pastoral intervention here. Their first child was born with Pater syndrome, which is one of the trisomies, an extra chromosome. And live births in Pater syndrome are extremely rare. And in fact, the consultant hadn't seen one before. They hadn't had any chromosomal or genetic testing during pregnancy. They'd only had scans, and they knew that something was wrong with the baby, but they nevertheless elected to go to term with it. They found it quite traumatic, having a baby that was immediately placed in an incubator with a lot of medical intervention, and it became apparent that unless that intervention carried on, he would die, and was going to die fairly soon anyway. I mean, Padhouse has got a live birth frequency of about 1 in 10,000. Its occurrence is more frequent than that because most of them die in utero. The consultant never seen one, but he got it right. He actually got it right. Roman Catholic, lovely man. And he supported the parents with this decision making too, but they eventually thought, let's just stop medical intervention and let little Hugh die. And the funeral was attended by a large number of members of our church and the many Muslims, because Lucy was the administrative assistant in our Department of Islamic Studies, which is very interesting, a Christian funeral for a little baby. They came to that. So she had a profound influence, but he had a profound influence. But whether, if you had asked him what he felt like, which you can't when he's 2 weeks old, it's a different question. It's also a different question for the little girl that John White talked about. You know, what did she feel about it? You can ask her maybe in heaven. But
maybe. Yes, I mean, simply sort of carrying on that conversation and then bringing out a little more, I think, the social context and impact can have on ethical decision-making processes and practice. And John, as you know, I think I worked in a prenatal diagnostic clinic for 5 years.
Indeed.
And there's a part of the world, you know, where family size is often between 8 and 12, and where a mother or parents would come to the clinic explaining that they watched maybe 3 or 4 of their children die slowly and painfully before the age of 8 or 9. The sort of disease that Tim was mentioning, like Lesch-Nyhan's, lysosomal storage diseases, those sort of things. Now, of course, in that particular culture, there was also the problem, especially out in the rural areas, that people felt that genetic disease of any kind, or a disability in a child, if it's slowly dying or was abnormal in some way, was seen as a judgment from God. And there were cases where that baby would be put in a dark cupboard and left to die because the sense of shame that the family felt, they felt guilty, you know, because they didn't understand what was going on.
Absolutely.
And so they felt guilty about this, and the baby was hidden away, you know, from public view and from view of the wider family. Now, it seems to me that making ethical decisions in those kind of contexts may well be different from in a context where some of the examples we've just been thinking about, where there was a very supportive family or where the community was around and supporting their child. It seems to me we can't make ethical decisions without taking into account, you know, the context in which
that's been made. And I think this is where absolutist views on these things can be unhelpful, you know, because they don't take into account the reality on the ground and so on.
I think I'm entirely with you on absolutist views, and there's a very fine book called The Moral Vision of the New Testament, written by an American conservative theologian whose name I can't remember, Richard Hayes, and he talks about how, you know, we need to transpose our Christian ethical decision making from those New Testament times when these problems were unheard of into 20th century America and Britain, writing in the 20th century, and talks about the importance. He uses abortion as a test case. He talks about the importance of that being a community, a set of virtues developed by the community, but nevertheless people will reach different conclusions. And so what right do we have to be absolutist about things for which we've obviously got no deontological guidance? We need to work it out in our communities, and I think what you've said is just an illustration of that in the community that you were referring to.
Switch this on. Do you want this on?
No, I just want to call up a slide here.
Yeah, do you want it to
No,
display?
it'll be all right. I just want to remind myself of something. Yeah, carry on.
I think that's a biological comment. Genetic diversity is the engine of evolution.
Yes, indeed.
We assume being reasoning human beings and etc., sort of at the top of the evolutionary scale. What are we doing for evolution now if we intervene so much? That's just a comment. I'm interested in Down syndrome.
Yes.
I'm not quite sure if you have a genetic test for it, whether it indicates how it's going to manifest itself, because it manifests itself across a wide spectrum. Would it be right to decide to abort when, in my experience, I know a Down syndrome young man who actually received an award in the southeast of England for the student that had made the most progress in a catering college. He now holds down a job. So he's at one end of the scale. If the decision is made, Down syndrome, we just don't allow an embryo to develop. There's a loss. What would ethics say in that situation?
Because Downs is not inherited but is sporadic, it's not subject to testing pre-implantation, but women who conceive over the age of 30 odd are automatically offered a prenatal test, and what they do with the data is up to them. You're quite right. What the data tell you is there's an extra copy of the particular chromosome, and the magnitude of the effect of that trisomy does vary from individual to individual, and this was emphasised to me long ago when I had my very first lecturing job after being a postdoc, when I was lecturing at the University of Nottingham, and there was a fine Methodist family whose youngest son was Downs, and he held down a job as a gardener in a horticultural concern and did it very well. With his instructions, he'd be out there, and he had a lot of support from the Christian community and obviously from his family, and that seems to account for a lot. The elder daughter and son-in-law of some very close friends of ours just had their second baby as Downs, and they're doing all the kind of support that little girl will need as she develops. But it's interesting that the case study that Richard Hayes took to discuss abortion in the Moral Vision Testament was Downs, and talked about different Christian couples reaching different conclusions. Most Christian couples I know in the UK would not abort on the grounds of Downs. Most of the ones I've spoken to, on the grounds that here you have a person who, with love and care and support, can make their way in life. But that's just my sampling of the Christian community. It might be different in the States.
Well, John, I think we should draw stumps out. Thank you.
Well, can I just give there's a Faraday paper called Ethical Issues in Genetic Modification. So if you go to the Faraday Institute website, it's there.
Yeah, they're in the back.
And also, if you go into the Centre for Bioscience Higher Education Academy, so that's That's www.bioscience.heacademy.ac.uk. There's a lot of ethical discussion papers there, some of them written by me. But there's one on pre-implantation genetic diagnosis, written by my friend Chris Wilmot, who's lecturing biochemistry at Leicester. And there's also one on stem cells, there's one on issues at the end of life, written by me and John Searle, for example. So they're written for secular context, of course, but they do raise some of the issues that society needs to think about.
Thank you. Just before we thank John, I think lunch is at 1 o'clock, isn't it? So we've just got a few minutes to browse the bookstore or gather your fresh air or whatever. So thank you again, John, very much.